much for the kind introduction and also for the opportunity that I can present our data here today. So I want to talk about our participation in the Endpoints project, which is involved in the establishment of new methodologies for endocrine disruption-mediated developmental neurotoxicity. So we want to develop methods which identify chemicals that harm the developing brain by interfering in hormonal pathways. During development, the brain is currently exposed to a variety of chemicals, and these can occur in air and water pollutions, in pesticides. They can be pesticides or food contaminants, or also pharmaceuticals. And when these chemicals harm the developing brain, they cause developmental neurotoxicity, short DNT. And consequences of DNT are neurodevelopmental disorders, such as intellectual disability, dyslexia, or for example ADHD or autism. And as one of these risk factors for developmental disorders, endocrine disrupting chemicals, short EDCs, have been identified. And examples here are bisphenol A, phthalates, or PCBs you've probably all heard about. And the problem is that for the hormone receptors we know that are expressed in the brain, only for some of them, here highlighted in green, we actually know how they influence brain development. But for the remaining ones, depicted in gray, we actually have only few clues how they influence neurodevelopmental processes. And that's why we actually have a concerning and dangerous data gap when we think of endocrine disruption-induced developmental neurotoxicity, and therefore also really insecurities for the pregnant woman. And that's actually when the Endpoints project was initiated, because the problem is that during the risk assessment process of EDCs, DNT is actually not tested in an endocrine mode of action. And that's why we thought, okay, we need new test methods which tackle this. And therefore Endpoints has 3 different tasks. The one task is to close this scientific data gap concerning endocrine disruption-mediated DNT. The second is the development of a testing battery, which mainly will comprise of in vitro new approach methodologies. And these methodologies shall detect chemicals that disturb brain development with an endocrine mode of action. And the third aim is to integrate these test methods into the regulatory frameworks. And what is our strategy at the IOF to tackle these tasks? So we are modeling neurodevelopmental key events in vitro, and therefore we use the test methods which are in the DNT in vitro battery you probably, or most of you probably heard about. And these include, for example, the proliferation of neuroprogenitor cells or their differentiation into neurons and oligodendrocytes and astrocytes. And what we do, we model these neurodevelopmental key events, and then we expose to hormone receptor modulators, so agonists or antagonists of hormone receptors. And once we identified neurodevelopmental key events which are sensitive to hormones, we go further to test method development. And for that we use the following cell model. We use human fetal neuroprogenitor cells, and they grow in these 3-dimensional neurospheres. And with these we can look at different endpoints. For example, we can look how they proliferate. We can differentiate them, and then you can see that radial glia cells migrate out of the sphere core. And when we have a closer look, you also see that they form nice cell-cell contacts. And when we give them 5 days to differentiate, you can see that they form a circular migration area. And in these we can then stain different cell types. In this case, we stained oligodendrocytes in turquoise and neurons in magenta. And modeling these key events, we then exposed to agonists and antagonists of hormone receptors and had a look how these events are actually affected. And here in this little scheme you can again see which number corresponds to which neurodevelopmental key events in the neurosphere assay. And in the lower part you see our output. We always generate concentration response curves, and from these we derive benchmark concentrations to compare effects. And this is a bit busy. This is actually the result of our screening. In the rows you see the different compounds we expose to. So we have for each hormone receptor an agonist and an antagonist. And in the different rows you see the different endpoints of the neurosphere assay, so the different neurodevelopmental key events we modeled. In blue you see effects where the hormone actually reduced or negatively affected the endpoint, and in red you see effects where the endpoint was positively affected. And all these effects were seen at non-cytotoxic concentration. We made that sure. The next step was to identify for each hormone receptor the most sensitive key event. And this is the event which is activated at the lowest concentration of the hormone. And for the receptors highlighted in blue, the most sensitive key event was oligodendrocyte differentiation, and actually we found that this neurodevelopmental key event was particularly sensitive to hormonal interference. In red, you see the hormone receptors where the most sensitive endpoint was neuronal differentiation, and in purple it was NPC proliferation. So the next step was here to see if there's also biological relevance, and that's why we compared our benchmark concentration we calculated with the fetal cord blood hormone concentrations. And for all but 2 receptors, we found out that our benchmark concentration we calculated are actually in the physiological range of the hormone. But for the progesterone E2 receptor and for the vitamin D receptor, we saw that our benchmark concentration highlighted in red were much higher than the fetal cord blood concentration of the corresponding hormone. So in this case, the effects we observed are maybe not biologically relevant. For the remaining receptors, we proceed in the investigation of the causal links between hormone receptor modulation and the affected neurodevelopmental key events we observed. And that's why we performed RNA sequencing analysis. And due to time reasons, I can't go into detail for all the receptors, but only will focus on 2 receptors. And the first one is the glucocorticoid receptors. And in the left scheme, you again see which endpoints were affected by modulation of the glucocorticoid receptor, and we observed that the most sensitive endpoint was progenitor cell proliferation. And what I did not mention before, we actually used neuroprogenitor cells both from male and female individuals, so we could actually also look at sex differences. And for the glucocorticoid receptor, we saw that male NPCs were much stronger affected. So when you activate the glucocorticoid receptor, proliferation is reduced, and especially when you look at male NPCs. The next step was to see if this effect is also receptor specific. And that's why we try to rescue the effect by co-exposure to the receptor antagonists. And you can see here that, again, dexamethasone, the synthetic glucocorticoid, reduces proliferation, shown in purple. And when we co-expose to increasing concentration of the glucocorticoid receptor antagonist, mifepristone, the effect was dose-dependently rescued. So proliferation was restored. Next, we then looked at our RNA sequencing results. And we performed the GO term analysis. And in light gray, you always see GO terms which were activated, and in dark gray, you see GO terms which were downregulated. And I want you to focus on the highlighted GO terms. We actually saw that a lot of genes are downregulated, involved in transcriptional processes. And we hypothesize here that glucocorticoids suppress NPC proliferation by negatively regulating transcriptional processes. And the second receptor I want to focus on is the liver X receptor, also a receptor which is not intensively stored before concerning brain development. And we saw that a lot of different neurodevelopmental key events were affected. And the most sensitive endpoint was oligodendrocyte differentiation, which was reduced. You can also see that in the pictures, that in the lower picture you see less oligodendrocytes staying for O4. Again, here we could confirm receptor specificity because the effect could be antagonized by the receptor antagonist. And when we looked at our RNA sequencing analysis, we actually saw 2 different possible modes of actions. We saw that on the one hand, lipid metabolic processes were deregulated, and it's important to know that oligodendrocytes are highly dependent on lipids, especially cholesterol, because they produce myelin, and for myelin production they need a lot of lipids, and disruption of lipid metabolism could be one reason for the diminished oligodendrogenesis we observed. And the second process we identified as deregulated was Wnt signaling, we saw that negative regulators of Wnt signaling were reduced, which vice versa then should mean that Wnt signaling is more active. And Wnt signaling has been already in the past described as a negative regulator of oligodendrogenesis. So also here we propose a second possible mode of action. When I sum that up in a little figure for all the receptors, you can see in this little scheme again the different processes which we can model with our different neurosphere assays. And when we have a look which processes are affected by which hormone receptors, we see that the glucocorticoid receptor and retinoic acid receptor impair NPC proliferation, and possibly by interfering in transcription and cell cycle. We saw that a lot of hormone receptors negatively affect the radial glia migration, and probably by interfering in the expression of adhesion molecules. We saw that actually several hormone receptors positively affect neurogenesis, and here we identified the Notch pathway, the Wnt pathway, and the mitochondrial complex 1 as possible modes of action. And we saw, as I already mentioned, that some hormone receptors negatively influence oligodendrogenesis, and here we identified Wnt signaling, MAP kinase signaling, and lipid metabolism. And from an old but previously published study, we already know that the thyroid hormone receptors positively influences the maturation of oligodendrocytes. The second step was then to see if we also can model these neurodevelopmental key events which we identified as hormone sensitive in other species, because we wanted to find out if the rat is a species which can recapitulate the effects we observed in human NPCs. And therefore we performed the neurosphere assays with rat NPCs because it was already established in our group, and we wanted to find out if the hormone sensitive key events are conserved and measurable using rat and neurospheres. And I want to show you a comparative picture of how a differentiated rat and human neurosphere look. You can see already that the different cell types exhibit a bit different morphologies, and our next step was to see if the hormone receptors are equally expressed between rat and human neurospheres. And what we also looked at on the left, you can see that we looked at expression in human cortical tissue of gestational weeks 6 and 12. And I want to highlight here that the expression pattern is quite similar. We see high expression of the glucocorticoid receptor, liver X receptor beta, PPAR delta receptor, retinoic acid receptor alpha, and thyroid hormone receptor alpha. So once we knew that the cells in general behave differently, we wanted to see if then for the most sensitive endpoint we identified using human neurospheres, we can recapitulate these effects in rat NPCs. And first looking at proliferation, where we identified if for human NPCs it was the most sensitive endpoint for the glucocorticoid receptor and the retinoic acid receptor, we could only recapitulate the retinoic acid receptor effect using rat NPCs. For the oligodendrocyte differentiation, we saw that some effects were we could recapitulate using the rat NPCs and some we couldn't. Actually, for the thyroid hormone receptors, interesting, we saw an effect only in the rat NPCs because oligodendrogenesis was improved, but not in the human NPCs. And for the neurogenesis, also there you can see that some effects we see the same across species and for some not. And we could conclude here that some neurodevelopmental key events are indeed regulated species overarchingly by hormones, but some are, to our experience, in a species-specific way. And for the hormone receptors, where we observed neurodevelopmental key events which were on the one hand in biologically relevant concentrations affected and on the second antagonizable, we proceeded to test method development. And only for one test method I want to go into detail. And if you want to know more about that later, more than happy also to talk about that later in more detail. And this testnet, I have to say, is already in pre-validation by PEPA, which is a public-private platform for the pre-validation of test methods for endocrine disruptors. And it is based on the effects I already talked about. So we observed that glucocorticoid receptor and retinoic acid receptor activation reduces proliferation. And we confirmed that this effect is receptor specific, so it could be antagonized. And what we also did previously, we excluded receptor crosstalk between the two receptors. And we found out that when we expose neurospheres to both hormones, so both to dexamethasone for the glucocorticoid receptor and retinoic acid for the retinoic acid receptor, so both hormones in combination reduce NPC proliferation even more. And then what's important, when we then, together with a hormone, expose to one of the receptor antagonists, we can see that each receptor antagonist is detectable in this setup where both hormones are present. And based on this, we developed a test method which can either detect EDCs which activate a hormone receptor by co-exposure to the antagonist, or EDCs that negatively affect receptor activation by co-exposure to the hormones. It is a two-tiered assay. And yeah, if you want to know more about the assay, I'm really happy to talk to you about that. And for the assay, we already established endpoint-specific controls, and we already tested the first EDCs and EDC mixtures. Yeah, and that leads me to my summary. So all neurodevelopmental key events were modeled, which are also part of the DNT in vitro battery, are regulated by at least one hormone receptor. We found that several hormone receptors regulate neurodevelopmental key events at physiologically relevant concentrations in the low and mid nanomolar range. Many key events are regulated by hormones in a species-specific manner. And here you can again see an overview about the most sensitive endpoint we identified for human and for rat neurospheres. And at the moment we are pre-validating EDDNT NAM for the disruption of glucocorticoid and retinoic acid receptor activity in proliferating neurospheres. Yeah, and with that I want to thank of course all the students that did a lot of this work. I want to thank my whole working group, and also of course the different funding sources and you for your attention. Thank you.
Thank you very much, Katharina, for your very interesting talk. So we have 5 minutes for questions. So if you have some, please go ahead. Maybe one at a time.
Yeah, that's better from you as well.
Yeah.
What is your predictability? I haven't seen that.
So the first question is, what is your predictability in the tests?
So you mean
You have set of test outcomes, right?
Yep. So at the moment we are still at characterizing our cell system. So we are not at the stage where we calculate the sensitivity or specificity of our test method. But for the neurosphere assay itself, it is already tested as part of the DNT in vitro battery. So we tested human positives and in vivo positives and human negatives, and then compared our testing results to the results from human and from in vitro studies, and there the predictability was actually quite high. But for these endocrine modes of action or endocrine test methods, we are not at the stage at the moment where we can already talk about sensitivity or specificity.
So my second question
Mhm.
is DNT assay. So as far as I understand, DNT is heavily influenced by the genetic factors and the environmental factors
Mhm.
together. Now you
Mhm.
are focused only on the environmental factors.
Mhm.
What is the method used to test the genetic factors in the assays?
Okay, so at the moment, you're right. We are really only looking at environmental factors, and actually that's also what our test method was developed for. We could, of course, we already established one model system in our laboratory for disease. And we can, of course, also model neurodevelopmental key events with using, for example, iPS cells with specific mutations in a disease-specific way. But at the moment, the neurosphere assay we are using for screening is only looking at environmental NOXA. So the question is which developmental stage is recapitulated by our model. And I totally agree, so it's important to keep in mind that our test method is only one part of a testing battery. And our test method recapitulates a certain stage of development. We have also further test methods which look at different neuronal subtypes, which also look at neural network formation and network function, which is of course not recapitulated by this assay. So this is a fetal developmental stage model, but we also have models which look at later stages. So it's always important to look at these assays, these in vitro nums, not only in the individual assay context, but at the whole battery of assays. You're welcome. So the question was if we also integrate our results in QAOPs for in silico prediction. So which is important to know is that in endpoints we have different partners, and one partner is completely involved in silico prediction and in QSAR generation, for example. So it's not our expertise in the project, but we have collaboration partners which are completely focusing on exactly that. So in silico prediction, and yes. Yeah, it will be also used exactly for that, together with the data from the other partners using different model systems. Yeah.
I have just one final question for you. So I saw through your presentation that you have a lot of vertical bars in the charts. Yeah. So, and since I'm coming from a metrology laboratory, I was wondering what that means for you. If you can explain if it is a variability, reproducibility of the tests, the variance. So in terms of quality demonstration of the results. What
Yes.
does that mean?
So the vertical bars you referred probably to that where I showed this antagonism, right? This exactly. So exactly. So this representation we only use for the co-exposures. For all the different exposures you saw in this big heat map, we performed concentration response curves, and also with best model fitting, and we derived benchmarks with confidence intervals. Exactly. So this representation with the vertical bars is only for the co-exposure. For all the benchmark generation, we actually looked at the confidence interval and make sure that the whole confidence interval also spans the benchmark response. So that's definitely important for us.